afternoon, everyone. Thank you for sticking around for those who have, or thank you for joining us. I may have mentioned this earlier, but my name is Ellen Holste. I'm the Community Program Manager at Pierce Cedar Creek Institute. So I'm really excited for our next talk. Our next talk is on Michigan's lost salamander, the Western lesser siren. This talk is brought to you by Adam Austin, who is a PhD student at Western Michigan University and also a research associate at the Rattlesnake Conservancy. So I'm really excited for your talk this afternoon, Adam. I want to take in all of your knowledge about sirens. I feel like this is such an unknown thing. And I'm pretty sure. Oh, go ahead.
It is so unknown, yeah. So there's so much we don't know about these species. So yeah, I'm really excited to share the story with everyone today. And it really is a story more so than it is a scientific presentation, because there's just so much we don't know about these organisms. But before I really dig into it, I want to go ahead and recognize my lab. I'm part of Dr. Tiffany Schriever's lab at Western Michigan University. So we study a lot of aquatic ecology, especially wetland ecology. And then we just ask this broad question of, how does environmental variability affect aquatic communities? So Morgan, she works in these rare imperiled interdunal wetlands, and she's looking at amphibian assemblages and plant communities. Sarah is another PhD student, pretty new to the team. She's working in river restorations and how aquatic communities respond to things like dam removals. And I'm working in mitigation wetlands. So a little bit more about my work, because I'm not maybe the most auspicious name here. I am a PhD student at Western. I am one of the instructors in the Department of Biological Sciences, and I study the community ecology of mitigation wetlands, so wetlands that have been created to offset damages to natural wetlands. A new project that I'm working on that I just started last year is I started working with the Rattlesnake Conservancy as one of their venomous handling instructors and one of the research associates. So they primarily have been operating out of the southern US with eastern diamondback conservation and translocation, but there's a growing interest for eastern Massasauga conservation. So we're starting to shift our focus that way. And we're really excited to offer a level 1 venomous handling course at Kalamazoo Nature Center, May 7th through 8th. And this is the first time we've offered one of these courses all the way up here in the Midwest. But the thing that I want to talk to you about today is a side project. It's one that, I did not plan on discovering or uncovering, and it is not part of my dissertation, and yet it is one of the most interesting pieces of research, I think. And that is about Western lesser sirens. So I'm going to start this story off all the way back in 1958. So there's a lot going on in the United States in 1958. This is the year that NASA was established. This is the year that the Russians launched Sputnik into space. Watson and Crick just published the structure of DNA a few years earlier. But in Van Buren County, a lake association was having a problem with common carp. So the carp that we see in the Kalamazoo River all the time and in these lakes, they're kind of a nuisance species. They're not native to here. And so the DNR decided to go out to the lake and apply rotenone. And rotenone is a pesticide. It's a pesticide that kills off fish and a lot of amphibian species as well, unfortunately. And they applied that to the entire lake to control that carp population. Well, a couple hours later, they received a call about some strange creatures that had just washed up dead on that lakeshore. And a biologist came out and identified both of those organisms as Western lesser sirens. And so John Williams, not the John Williams, the composer, but John Williams, who is working in the Institute for Fisheries Research in the Michigan Department of Conservation, now Michigan DNR, published this distribution note to Copia, where he also said, the Western lesser siren may have been discovered and made extinct here at the same time. So these two sirens found at this lake in Van Buren County was the first time we had ever found them this far north or in Michigan. Previously, the furthest north that we had found them was central Indiana. So this was a really interesting discovery, and they were very curious about how those wound up here. Fortunately, though, 5 years later in Allegan County, so only about 10 to 12 miles away from that original site, once again, they were applying rotenone to the water to do a study or control of fish populations. We're not quite sure which, but they found a single dead Western lesser siren and reported it then. And this observation, all the way back in 1963, so 59 years ago, is the last time that we had a recorded observation of a western lesser siren in Michigan. Since then, there have been anecdotal accounts, but there's never been photo evidence, there's never been a body produced, and even those accounts have been few and far between. Now, I'm going to pause the story right there real quick because I want to talk a little bit about what western lesser sirens are, because it occurs to me that they're very rare here in the Midwest and in Michigan especially, and most people might not be familiar with them. So western lesser sirens are Siren intermedia. They are neotenic salamanders, just like mud puppies. So that just means they retain these larval traits like these bushy gills into adulthood. Some other examples of neotenic salamanders are things like oxalotls, mud puppies for sure, but most other salamanders outgrow this larval phase. They go through metamorphosis, just like tadpoles do into frogs, and they become terrestrial. Lesser sirens stay fully aquatic for their entire lifespan. They do have these bushy external gills. They also have lungs, so they can go up to the surface and gulp air and breathe, so they can survive in very low oxygen environments as well. One of the notable characteristics is they have these very long eel-like bodies. And in fact, if you didn't see the head, you would think that you're looking at an American eel or something very similar to that. They have two small front limbs. And the reason I mentioned that they just have two small front limbs is they don't have any rear legs at all. They don't even have a pelvic girdle or vestigial hips. So the lineage of sirens taxonomically is really ancient. And it looks like in this lineage, tetrapodomorphism or four-leggedness was something that never really developed. And they can be distinguished from other siren species by the costal grooves. So counting along the rib cage, they have usually less than 35. The other larger sirens in the South have more than 35. So their behavior and ecology, and the top you can see a juvenile lesser siren. They're very brightly colored when they're young, and they lose a lot of that coloration into adulthood. They are a nocturnal species of salamanders, so they're primarily active at night. And during the day, they are hiding under debris at the bottom of lakes and ponds, or they burrow into the substrate. They breed in the very early spring, right? So they practice external fertilization, so laying eggs and fertilizing them within these debris-covered cavities. They have a really complex courtship behavior that we don't understand very well. And the reason we don't understand it is we've only observed it ever happening once in captivity. So a lot we don't know about these sirens and how they breed. They do grow quite quickly. They go from, being about an inch or two long at that juvenile phase to adult size, which can be about 24 inches, and they can grow that large within 1 year. So they do reach a sort of a size refuge from predators very quickly. And they have this really thick slime coat. So if you've ever tried to handle an eel or something like that, and you just can't get a grip on it, that is what sirens are like. So it's not like a frog where you can catch it and yeah, it's slimy, it's kind of slick, but sirens, you literally cannot grip onto them. And they use that slime coat to prevent infections, to keep them from drying out. But they also are able to do something that we also know lungfish can do, which is if a pond completely dries out, sirens can create this excess slime cocoon. And then they can survive for months without water just hidden in the soil, even up to a year or two, just in those conditions. So their diet and feeding, they primarily feed on invertebrates or small vertebrates if they can find them. And they utilize suction feeding. I think I have a short video here. Hopefully that and the audio works for you. That just shows some of this feeding activity. They have really poor eyesight. They can tell whether it's day or night, but they can't really see much going on in the water. So they rely on chemoreception or smell to detect their prey. Okay, so their habitat and their range. If you look at this range map, which I stole from iNaturalist, so these are iNaturalist citizen science observations. The lesser siren is really congregated around the Gulf coastal plain. So down into Louisiana and the Mississippi Delta along Texas, and then they kind of follow the Mississippi Basin and then the Illinois River up into the Midwest, and they become very scarce. And then, of course, you have the observations in Southwest Michigan from back in the 50s and 60s. Unlike mud puppies, they really prefer very dense aquatic vegetation and very soft substrate. So the type of lake bottom or pond bottom that would cause you to sink into it, that's what mud puppies, or not mud puppies, I'm sorry, lesser sirens prefer. Yep, they do range along the Gulf coastal plain up into Mississippi. But the big unknown is if they still occur in the Great Lakes Basin. If they occur in Michigan, then they do. If they're not in Michigan anymore, then they don't. And so it brings us to their status. In the South, they're fairly common. Their populations are declining, but they're still fairly numerous. You can still find them in a lot of these lentic water systems. But in Michigan, again, we haven't had any records of them since 1963. And while they used to be on the endangered species list, they were downgraded to a species of special concern. And this question that we've had for the past 50 to 60 years is the western lesser siren extinct in Michigan? And there were some strong pushes to just have it declared as extirpated so that we're not devoting any resources towards a species that's only here in theory. So this kind of brings us to the modern day. I had one of these funnel traps just sitting out on my dock at my lakefront property, and I left it in the water for a couple days longer in the middle of August than I probably should have. And I pulled it up, and as is tradition when it comes to discovering sirens, it was dead. And I didn't realize how rare these species were or that it was such a significant find. So I felt really bad about it. I said, that's a shame. I tossed the body back into the lake. So I didn't have a photo. I didn't have a body to present. And naturally, the DNR and Dave Mifsud were a little skeptical when I said, we found something that hasn't been found in 60 years. And so for the next year or two, I set out on a kayak every day when I got home from work. And I spent about an hour and a half going around the lake and checking about 20 of these different traps scattered in and throughout the weeds. And so we've had some results.
We're back at the same location as last year. Actually the same trap out here. And we've got another western lesser siren. Today is the 27th of March. It's March 30th, May 13th in our second location. Out here once more. So this is our second one this year. Looking at our third lesser siren for the year. We just finished getting our DNA sample, so we're going to go ahead and just let this guy go real quick. Might be better just
Right. So after about 3 years, initial discovery in 2018, and then a season in 2019, 2020, and 2021, and spending hundreds of man hours, going out and checking these traps in the early spring, and then trying to do it throughout the summer, we have managed to find 6 sirens. So they are here. They require a lot of effort to find, though, as you would expect with a species that's fully aquatic, nocturnal, and prefers to hide in the substrate all day. They're very difficult to sample for. So if you're trying to sample for them, if you're trying to survey, you have to go out in the early spring. As soon as the ice melts off the lake, that's the best time to find them. I found that once the vegetation starts to grow in, they become almost impossible to locate. Some people in the past have used electroshocking and in fact have not found sirens until they used electroshocking. Seine netting is another option. So another photo up here on the left where you drag these big seine nets through the water and whatever comes up, comes up. And then of course, funnel traps like Dr. Greenwald was describing. So we actually tried to do electroshocking because the funnel trap was very labor intensive, and it looked a lot like this. They do prefer those really soft substrates, and so This is more or less a reconstruction of what happened back then. We were buried in the mud with an electroshocker, and it really didn't work out great. Again, you can try seining through these systems where you have this deep muck and mud, but then again, you're fighting vegetation and the sirens burrow into the substrate. So if the seine line isn't taking all the substrate with it, you're going to pass right over them, and you're still going to have a problem very similar to this. So we decided funnel traps were the best method. That's what we're still using now. And so what do we do with these sirens when we collect them? Well, the first thing we do is we put them in an anesthesia bath of MS-222, just to get them to kind of settle down. You can't grip them, so you don't want them moving around while you're trying to take measurements. The measurements we take, we measure the snout to vent length, we measure the number of costal grooves, the head width, the head length, and the girth around the middle of the animal, just to try to detect any differences in morphology. We take a toe clip for DNA samples, and toe clips in these salamanders actually do grow back. And then to make sure that we're not sampling the same sirens again and again, we take an 8mm biomark pit tag, so these passive integrated transponder tags that you see here, and then that just goes right into the muscle of the tail right behind the cloaca. We wait for them to recover from the anesthesia, and then they are released right back at the capture site. So we're collecting all this data because we have a few questions. And one of the obvious questions is, how widely distributed are sirens in Michigan? If it takes hundreds of hours of sampling, how, there might be a chance that they could be more widely distributed than we expect. And in fact, we have some unconfirmed reports of them existing as far north as Muskegon. We want to know when and how they first arrived in Michigan. Were they brought in by an angler, or did they disperse into Michigan through some natural corridors? We also want to know if they're distinct from southern populations. The climate and the environment in Michigan is substantially different than the Gulf Coastal Plain and the swamps of Louisiana and Texas and Oklahoma. So are they different in their morphology, their physical characteristics? Are they different genetically? Do they exhibit certain behaviors? And really, how can we conserve that population? Western lesser sirens are significant for a few reasons, and like Dr. Greenwald said, they're pretty cool. And most of these aquatic salamanders are just really neat, especially if you're a herpetology enthusiast. They also control some invertebrate populations. They're an important invertebrate predator. They're also a good food source for predators. A lot of herons and larger fish species will eat sirens as well, so they serve as a link in that food chain. They are this ancient lineage that doesn't have tetrapodomorphism, so studying them can help us understand the evolution of amphibians and vertebrates in general. And they could potentially be a new species or subspecies. They've been isolated for long enough. They could have unique characteristics that distinguish them from their southern counterparts. And the reason I say this is in 2018, so just a couple of years ago, Sean Graham described a brand new species of siren in the Gulf Coastal Plain called the reticulated siren. And almost everyone who has written research on sirens has said, we know so little about them, we are probably going to discover new species and subspecies. And overall, we really know nothing about these animals. They're very hard to observe in the wild. We don't have a lot of captive observations of them. And they're really poorly known how widely distributed they are. They face a number of threats right now. Habitat loss is a threat for pretty much every amphibian, but also for sirens. Persecution or collection. This is more for the southern United States. Sirens have been used in the past as fishing bait. They're also collected for the pet trade. Industrial chemicals, so runoff from fertilizers and pesticides and different herbicides. And I have dicot on here, and I have this asterisk in balancing nuisance vegetation control with siren conservation, because on the lake where we found these sirens, it was only 2 years after we rediscovered them that the lake residents were fed up with the amount of vegetation in the lake. They were really tired of constantly having to cut and rake their beach. So they wanted to contract dicot to be applied to the entire lake. And dicot can have these mutagenic effects on salamanders. It affects their metabolism. And we really don't know the direct effects that it might have on sirens themselves. but also killing off all that vegetation at once usually causes nutrient spikes, and it can remove shelter from some of those large predatory fishes so that they can more easily pick off these animals. So how can you help? How can we conserve these species? There are a few ways. One is protecting their habitat. So if you have lakefront property or you are managing ponds, allow a vegetation buffer around the lakefront. So do a little bit less work. You don't have to necessarily plant a garden like you see here on the right, but you can just avoid mowing, within about 10 meters of that lakefront. Avoid applying any chemicals that could affect these species, so pesticides or fertilizers for your lawn. And if you're going to rake your beach, which is fine, just make sure to sort through the debris. Already residents on our lake have accidentally raked up sirens along with the vegetation, and fortunately they were okay and they were able to be released. But if you have a large aquatic weed harvester, that can be a really dangerous situation for these animals. We also need to know where they exist, so we need documentation of their range. So if you observe them at all, please report them to the Herp Atlas, and remember to take a photo, unlike me, or iNaturalist. iNaturalist is one that I consult often. It is, unfortunately, because they're a species of special concern, they're not legal to intentionally trap or net without a scientific collector's permit. However, if you do not intend to catch them, but you happen to have a funnel trap out, or you happen to be seining and one happens to end up in your net or happens to end up in your trap, that's totally fine. Grab a photo, upload the observation, make sure to release it on the site, and that will help us out a lot. We are also interested in any kind of funding or interested researchers for this. Again, this is my side project. This is not my dissertation. There's nobody else collaborating or involved with this other than the folks that I'll list in the next slide. So it is ripe for grad student projects or undergrad honors thesis. If you have funding opportunities or know of funding opportunities, we'd love to hear about those as well. I want to go ahead and thank Dr. Tiffany Schriever and Dr. Devin Bloom for all the material support they provided me and the mentoring throughout this project. I've had some really great conversations and insight with Dave Mifsud, also Dr. Bob Broadman, who researched sirens in northern Indiana, and Jim Harding as well. And I also want to thank again Dr. Tiffany Schriever and my lab mates Nicole Stewart and Morgan Warren for helping out during fieldwork. And at this time, I can take any questions.
One such question, and Katie, I had the same question. So Katie Greenwald asked, what molecular work are you or others doing with the DNA samples? Just curious about differentiation between other distant populations, and seems like a great opportunity for eDNA. That was my question too.
So you know what? It's an awesome opportunity for eDNA if we're trying to figure out geographically where they're distributed, and that can be a really helpful tool. The only issue with eDNA is that it doesn't necessarily pinpoint the locations where they're coming from. A lot of these water bodies are very interconnected, and we kind of want to know those microhabitats. But we're also very interested in, how they compare to southern populations. And eDNA is great for that, but we really need that pure tissue sample and those morphological measurements to sort of answer those questions. But eDNA can be an important part of this toolbox that we use to look into them. As far as what molecular work we're doing, Sean Graham did his molecular work when he described the reticulated siren. So he has a list of primers. I believe there's some stuff in GenBank we can use, but for the most part, these tissue samples are being stored in ethanol in minus 25 degrees Celsius freezer, and we're kind of sitting on them. So like I said, there is no funding on this. Is kind of on my own time. So we really haven't moved forward with the genetic analysis component yet.
Okay, good to know. Somebody did ask, do sirens also like cheese? Since we just talked about mud puppies liking cheese as another way to trap.
I have not attempted it. I have tried baiting traps before. Unfortunately, when you bait traps in these environments, you usually end up with a lot of predatory fish like bullhead or pickerel in the traps, or even some of the larger predatory invertebrates like the Lithoceros giant water bug species that could potentially kill them off.
Yeah, and that was a good point. When you were talking about the eDNA, it made me think you talked about dense aquatic vegetation as their preferred habitat, and then this interface with humans and our environment, where what one species wants, another may not. So yeah, no, great inspirational talk. If you have any other questions, we still have time for questions, so feel free to put them in the Q&A or the chat. If you're okay, Adam, I will also share your email to if people have other questions that they think of in the future. I know this is a very ripe project, as you said, for future research. And so students out there, professors out there, this is good to go. So yeah, and as Kathleen said, this is a great example of one person that can make a difference, and a big difference. So thank you, Adam, for all your work on this, and this being your side project. Regardless, it's inspirational to know that things that we thought weren't there anymore are actually still there.
Well,
So.
we've just got to look. Thank you so much for having me.